We're going to start off by drawing a line across the bottom third section of the page, and then we're going to draw a chloroplast. The chloroplast is a double membrane organelle, so I'm also going to draw a second membrane. Then we're going to label the parts of a chloroplast. Then we're going to draw a thylakoid. We're going to draw a big thylakoid. It's going to be where we see a lot of these important reactions occurring. So there's my thylakoid, and we're also going to write down the light-dependent reactions, as well as the Calvin cycle, which would be the light independent. Okay, now we're going to draw the photosystems, and we have two photosystems that I'm going to draw looking like bell peppers here. Let's make this a little bit bigger so we can see what's going on. This is photosystem 2 because it was discovered after photosystem 1, so we're starting with photosystem 2. Next, I'm going to draw an electron carrier that I'm going to abbreviate PQ. PQ stands for plastoquinone, and this is a electron carrier. I'm also going to draw a cytochrome complex and a protein called plastocyanin. Next, we are going to draw our second photosystem, which is photosystem 1, because this one was discovered first, this one was discovered second. And then I'm going to draw a carrot-like shape. This is our ATP synthase. Within a photosystem, there are two complexes. One is called the reaction center complex, which is going to be just in this center region over here. And each of these photosystems has one. Within this reaction center complex, within each one of these, there are two important structures. You have a special pair of chlorophyll, which is, in this case, is called P680, referring to the wavelength that the special pair of chlorophyll molecules is the best at absorbing. In photosystem 1, the special pair of chlorophyll molecules is P700, again referring to a wavelength of 700 that is best absorbed by photosystem 1. These components, this square region, is called a primary electron acceptor. That is capable of being reduced. And we'll see how it becomes reduced in a second. Now we're going to be looking at another part of the photosystem. So every photosystem contains pigments. These pigment molecules make up what is called the light harvesting complex. And this one I'm going to label here. So there's all of these pigments where photons of light are going to be able to bounce around. when light hits these photosystems. Okay, let's start by putting an electron. So this electron here, it is currently in what is called a ground state, meaning that it is in its most stable form. A photon of light is going to strike one of these pigment molecules. It's going to excite an electron, which is going to excite another electron in another pigment and excite another one, exciting all of these pigment molecules until it reaches this special pair of chlorophyll molecules called P680, where there is an electron. This electron, currently in its ground state, is going to become excited. Once it does, it's going to be accepted by this primary electron acceptor. So now the electron is up here. It has gained potential energy, and this electron now has more potential energy than it started with. It is going to be able to feed through this electron transport chain and enter this P700 special pair of chlorophyll, where it's going to wait for another photon of light to come in and transfer its energy from one pigment molecule to another, eventually giving the energy to that electron that will then be boosted to a higher energy level and accepted by another primary electron acceptor. So this electron now has gone through 2 rounds of excitation, and it contains a lot more potential energy than it had started with. All right, here we're going to draw a protein molecule. It's called ferredoxin, abbreviated FD. So by moving through ferredoxin, the electron is actually moving through a second electron transport chain over here. The photoexcited electrons are going to travel through the protein ferredoxin, and an enzyme called NADP plus reductase is going to help reduce NADP plus to make it NADPH. So it takes 2 electrons to reduce NADP plus into NADPH. One way that I like to remember the difference between NAD plus and NADPH is that NADPH has a P, which I associate with photosynthesis. NADP plus is an electron carrier. It's very similar to NAD plus. The only difference is that there's an extra phosphate group, and that's why it's NADP instead of NAD. So this gives us the first product of the light-dependent reactions, NADPH. I'm going to write the products down here. Actually, I'm going to write the inputs and the outputs. We have NADPH, and as these electrons were moving down this electron transport chain, moving through plastoquinone, the cytochrome complex, and plastocyanin, the electrons were they were releasing some of their potential energy, and the released energy is going to help pump protons across the membrane of the thylakoid. In this case, there are protons just in the stroma, and these protons are going to be pumped into this thylakoid space. So an accumulation of these protons is going to generate a proton gradient, which is going to become a proton motive force that will help run ATP synthase to produce ATP. And how that's going to happen is that these protons are going to want to move from an area of high concentration to an area of lower concentration. So when these protons diffuse through ATP synthase, they are going to assist in the phosphorylation of an ADP with a phosphate group, which makes ATP. This is the second product of the light-dependent reactions. So ATP, and these two are going to go into the Calvin cycle. But before we get there, there's another product, more of a byproduct of this light-dependent reaction. And let's go back here to these. So let's go back here to this electron. Once this electron has been excited, it is going to leave this chlorophyll, this special pair of chlorophylls, and we're going to need another electron to take its place. So water, this is where water comes in. Water is going to, let's see, where can I draw water? Hold on. Let me erase that label. Just rewrite it right here. Photosystem 2, that way I can draw a water molecule over here. So a water molecule, I'm going to draw out the whole 8 electrons and the hydrogens. So a water molecule is going to split in half, which is going to generate 1 oxygen atom, 2 protons, 2 electrons that it's going to feed into this now empty chlorophyll space. So that when another photon of light hits this light harvesting complex, it's going to be able to photoexcite this electron so that it becomes excited and also goes down this electron transport chain where it's accepted by another special pair of chlorophyll, since this would now be empty, since the other one had already been photoexcited and has gone to reduce NADP+. So this electron would then be phosphorylated. So this electron would then be photoexcited once again and become part of NADP+. So that was a quick overview of what basically happens to these electrons. And we were on the topic of how these electrons are provided to these special pairs of chlorophyll. So they come from water. Water donates its electrons into photosystem 2. It splits up into all of these components. The protons remain in this thylakoid space, and the electrons are what's fed into the chlorophylls one by one, and they get excited one by one. The oxygen, on the other hand, is just 1 oxygen atom, so it takes 2 water molecules to make 1 oxygen molecule. Otherwise, 1 water molecule would only make a half of an oxygen molecule, because it only has 1 oxygen atom, and you need 2 to make a diatomic oxygen molecule. So oxygen is another, we're going to put it under outputs, and here we're going to put water as the input, and of course light. And I put in parentheses a photon of light. Okay, so we know that water need 2 of those to make 1 oxygen molecule. We know that ATP is made because of the electrons that move down the electron transport chain towards NADPH, towards NADP plus, to make NADPH. And in the process, the protons are what helps make ATP, that proton motive force. Okay, so another thing I want to point out is this over here is pretty much chemiosmosis, which is very similar to the one in cell respiration, where a proton gradient was made from the electron transport chain. Protons were pumped in, there was a high concentration on one side compared to the other, which led to this overall diffusion, this movement of molecules from one side of the membrane to the other side. And in the process, phosphorylating ADP to make ATP. Okay, so the next part of this photosynthesis process, this is the light reactions. Second part is the light-dependent or dark reactions, or the Calvin cycle. We know that NADPH and ATP are the inputs into that cycle. So we're going to look at how exactly they are input and when they come in. So I'm going to draw a cycle, just kind of like this. That might be a little bit too big. Hold on. Make it smaller because we have some stuff to write around it. Okay, so an input into the Calvin cycle is carbon dioxide. And the Calvin cycle is very similar to the citric acid cycle or the Krebs cycle in cell respiration, in that it is a cycle and it regenerates a specific molecule with every cycle. That molecule is a 5-carbon, let me just put C's inside so we know they're the carbons. So this is a 5-carbon molecule called RuBP, and that's short for ribulose bisphosphate. And for every cycle, RuBP is going to join with the carbon in carbon dioxide. So I'm going to draw carbon dioxide looking like this, with its 1 carbon atom and the 2 oxygen atoms. The reason why I'm drawing 3 here is because, let me skip ahead to the products that we are expecting. So we know that we commonly associate, photosynthesis makes a glucose molecule. Glucose is a 6-carbon molecule, but it's not as simple as that. The direct product of the Calvin cycle is actually a 3-carbon molecule called G3P, and this is short for glyceraldehyde 3-phosphate. So the direct conversion of these carbons, the direct conversion of these carbon atoms, so the direct fixation or integration of these carbon atoms from carbon dioxide makes 1 G3P molecule. So you can see it takes 3 carbon dioxide molecules to make 1 G3P. Each one of these carbon dioxide molecules is going to join with 1 RuBP, which means that for every Calvin cycle, there's actually 3 RuBPs, 1 for each of these. And what this makes, the combination of RuBP with a carbon dioxide molecule is going to make a 6-carbon molecule. Hold up, it's not glucose. This is 6 carbons. This is a very high energy molecule, so high in energy that once it's created, it actually immediately splits in half. So it splits in half. So we're just going to follow the path of just one of these now, because I don't have enough space to draw all of them. We're just going to follow the path of, let's say, this one. Okay, so this one integrated with this RuBP makes our 6-carbon molecule. It is split in half, so it becomes 2 3-carbon molecules. For every carbon dioxide, it's going to eventually make 2 3-carbon molecules. All right, so this process is called carbon fixation. The fixating, the integration of carbon dioxide into this Calvin cycle on the path to making a sugar. Okay, it's going to combine with RuBP, become a 6-carbon molecule that is going to split in half, becoming 3-carbon molecules. So once it's become these 3-carbon molecules, we're just going to multiply this by 3, since, this would happen, this is the first time, this is the second time, this is the third time. So we're just going to multiply like this by 3. And we're going to go on to the next step of this process. So number 2, the second step is called reduction. The reason this step is called reduction is because 2 things are going to happen. First, one of our products from the light-dependent reactions, ATP, is going to give energy into this cycle and become adenine diphosphate, so it's going to lose one of its phosphate groups. Okay? And then another process that's going to happen, I'm just running out of space, so let me just reposition my G3P. So another process that's going to happen is that the other product, NADPH, is going to become oxidized, meaning it's losing or donating its electrons into these carbon skeleton molecules. The process becoming oxidized, NADP+, that's the oxidized form. So here, let me just put a square around the oxidized and the dephosphorylated forms. This one is what's donating the electrons. So these 2 processes that take place are in the reduction phase, and they ultimately help make G3P. So this 3-carbon molecule is the product of reduction. So technically, since there are these 2 times 3, there's 6 of these G3P molecules being created. The reason why we put the product, the most direct product, as 1 G3P is because only one of these 3-carbon molecules is going to eventually become integrated into glucose. And since glucose is a 6-carbon molecule, it takes 2 G3P molecules to make 1 glucose molecule. What happens, you might be wondering, there's 6 right here. Why does this cycle have to, repeat itself? Why can't we just use these 6 and make, 3 glucose molecules? There is a third step called the regeneration of RuBP. Should give you a hint. So remember, RuBP here is a molecule that is constantly regenerated in the Calvin cycle. Its main purpose is to fixate carbon dioxide from carbon. Its main purpose is to fixate the carbon from carbon dioxide into this very high energy 6-carbon molecule, breaking in half, and of course with reduction it is going to become G3P, and we only need 1 of those so far. So to regenerate RuBP, we actually need 5 of these G3P molecules. Remember, one of them has gone off to become glucose, and the remaining 5 are going to become reorganized in a way to regenerate RuBP. Alright, so pausing here to do a quick recap of the carbons. We're going to follow the carbons through the Calvin cycle and keep track of how many carbon atoms we are actually working with. So we have 3 carbons from the 3 carbon dioxide molecules. That is going to combine with 3 RuBP molecules, each one containing 5 carbons. So that's 15 carbons from RuBP plus 3 carbons from 3 molecules of carbon dioxide. So that's 18 carbons. And that's going to make 3 molecules that contain 6 carbons, and those are very high energy carbon molecules that are going to split in half and create a total of now 6 molecules of 3, a total of 6 molecules, each containing 3 carbons. Now that still equals 18 when you multiply 6 times 3. Then reduction, in the reduction phase, they become G3P molecules. So you have 6 G3P molecules. One of them is going to leave to become the precursor to glucose. The remaining 5 G3P molecules are going to total 5 times 3. So 15 carbons are comprising the remaining G3P molecules that will become regenerated into 3 molecules of RuBP. So after a G3P molecule leaves, we now have 3 less carbons. So we have 15, and those 15 come from 5 molecules, which after regeneration and reorganization is going to become 3 molecules of RuBP. So the 3 molecules of RuBP, again, they contain 15 carbons total. They are ready to accept another 3 carbons from incoming carbon dioxide, and the Calvin cycle will then repeat. So you have a total of 18 carbons after carbon fixation. And during reduction, after reduction, you have lost 3 of those carbons in the form of G3P. The remaining 15 are going to be recycled and regenerated into RuBP, which is going to accept another 3 molecules, making 18 all over again until another G3P molecule leaves. So this is just a quick review of the carbons. Hopefully you can better visualize and see the path of these carbon atoms in the Calvin cycle. Hopefully with this math it makes more sense. And then we're going to continue with the addition of ATP into the Calvin cycle to regenerate RuBP. And this is a process that actually takes up some ATP as well. So ATP is invested in this regeneration process. Okay, so we can see our inputs and our outputs here now. Our inputs from the light-dependent reactions were NADPH right over here in this in the reduction phase, as well as ATP. This was used in both the reduction phase as well as the regeneration phase. And another input here is carbon, and that was in the carbon fixation phase. So I'm just going to write a little note as to, actually, you can write that down if you want, the exact phases in which each of these inputs were used. From here, I'm just going to write the inputs and the outputs. So we know that NADPH becomes oxidized. It loses its electrons to what's going to become a G3P molecule. So it becomes NADP+. Also has an extra proton here. ATP becomes dephosphorylated, becoming ADP with another phosphate group. Carbon dioxide becomes fixated with RuBP. Ultimately, after several steps of carbon fixation and reduction, becoming G3P. And we need 2 of them to eventually make glucose. Since it takes 2 molecules of G3P, and for every Calvin cycle, you make 1 G3P, that means you need this Calvin cycle to run twice in order to make enough G3P molecules to make 1 glucose molecule. So now I'm going to show you the stoichiometry behind everything on a different page since I don't have space here. All right, now for the numbers. It takes 2 G3P to make a 6-carbon glucose molecule. That's because G3P is only a 3-carbon molecule. We would need 2 of these to make a 6-carbon glucose molecule. So if G3P we need 2 of, and the Calvin cycle only produces 1 G3P molecule, for every cycle we would need the cycle to run twice, meaning that all of these inputs we'd have to multiply by 2, including that one. Okay, so I'm going to write down these inputs on the side again. So there's 3 main inputs, starting with, let's look at ATP. So there are 2 inputs of ATP. They total, so this one is an input of 6 ATP, and on this side it's an input of 3. So there's a total of 9 ATP required to generate 1 G3P molecule. We're just going to ignore this right now. So 9 ATP is required, and we need 6 NADPH for every 1 G3P molecule. We also need 3 carbon dioxide molecules. That one we already had written down earlier. And this is all just to make 1 G3P molecule. So if we need the Calvin cycle to run twice, we have to multiply everything by 2, which means we actually need, a total of 6 carbon dioxide, a total of 12 NADPH, and 18 ATP molecules. And remember, these 2 come from the light-dependent reaction. So we're going to look over here and just work backwards to make sure we account for all of the inputs and the outputs of the equation for photosynthesis. Okay, we got 1 glucose molecule, and that is going to require 6 carbon dioxide molecules, and these 2, which are products of the light-dependent reactions. All right, so we know that for every water molecule that is broken, let's actually start with 2 water molecules. So for every 2 water molecules, you can make 2 NADPH because you have 2 pairs of electrons. From 2 water molecules, you have 2 pairs of electrons, and each NADPH molecule carries 2 electrons. So you can make 2 NADPH molecules, and then remember there are protons that are pumped across the thylakoid membrane from the stroma into the thylakoid space because of these electrons moving down their electron transport chain. These protons that are pumped across the membrane, we will have 12 of for every water molecule. And if you remember the conversion between how many, and if you remember the amount of protons it takes to generate 1 ATP molecule, remember going through ATP synthase, it takes 4 protons to run this rotor that is going to make 1 ATP molecule. So just doing the math here, we would get 3 ATP molecules for every 2 water molecules. That releases 4 electrons into the electron transport chain, pumping 12 protons into the thylakoid space. All right, so this is where the ATP comes from. And we know that for every 2 water molecules, we make just 1 oxygen molecule. Remember, oxygen is a diatomic molecule. You need 2 oxygen atoms to make 1 molecule of oxygen. We know we need 12 NADPH and 18 ATP. Here we have 2 NADPH and 3 ATP. What is the number you would have to multiply everything by to get 12 NADPH? How many times would you need this to run to generate 12 NADPH? Right, so 12 divided by 2, you would need this to run 6 times. So you would need everything here in this circle to run 6 times. You would also need 18 ATP. Here we have 18 ATP. If we multiply 3 for every cycle times 6 of these cycles, technically not cycles, but reactions. 6 of these reactions, you would get 18 ATP, and so far this is consistent with what we need to make 1 molecule of glucose. Now looking at water here, remember we needed 2 molecules of water to make all of this. So when we multiply everything by 6, you're actually multiplying this water, that's already a times 2, times 6. So this gives us actually 12 water molecules input into the light reactions. So when we write out the equation for photosynthesis, I'm going to write it out up here. See if this is consistent with everything that we know. So 12 water molecules input, obviously plus light. Here we had, let's look down here, 6 carbon dioxide molecules. It's going to give us, we got our oxygen and we got our 1 glucose. So our oxygen here is also multiplied by 6. So we get 6 oxygen molecules. Remember this is, everything in here is multiplied by 6 so that we can get the appropriate values we need for NADPH as well as ATP. So oxygen here also is multiplied by 6, hence we have 6 oxygen molecules produced plus 1 glucose molecule. And glucose, I'm going to write it out as this. Okay, so this is our equation, and we're actually missing something over here. So I'm going to write 6 water molecules. This is not mentioned, at least in our textbook. The Campbell textbook doesn't mention the uptake, or the Campbell newest edition for AP Bio doesn't mention the production of 6 water molecules in this process. But I want you to know that this happens somewhere. Somewhere, somehow, 6 water molecules were also produced. So here we have 12 water molecules input, 6 water molecules produced. Usually you see the equation for photosynthesis as a net equation. So meaning that the production of water molecules was taken out, so minus 6 water molecules. Got to balance the equation. Take out 6 water molecules from the input side. That gives us 6 water molecules plus light plus 6 carbon dioxide molecules makes 6 oxygen molecules and 1 molecule of glucose. So this equation here, I am going to rewrite it again. I'm going to leave out light this time. We know light is part of it, right? Whoops. So this equation that you commonly see is the net equation for photosynthesis, meaning that it's already taken into account the amount of water that was produced here, and this is the net input of water. So taking into account that there was already a production of water, and just to simplify this equation, they only mention the net input. Okay, so we got water, carbon dioxide, oxygen, glucose. We know there's also light. And there it is, our equation for photosynthesis. So hopefully now you know how the inputs become the products, how many of each of these inputs makes the products that we expect. And ultimately, I hope that this was a helpful visual for you in understanding the process, the light-dependent reactions, and the Calvin cycle, dark reactions, the light-independent reactions. And for the Calvin cycle, all you have to know is the 3 main steps: carbon fixation, reduction, regeneration of RuBP. Obviously, this is the Calvin cycle. Knowing the inputs, meaning the products from the light-dependent reactions, as well as carbon dioxide, that is input to make these products that will be recycled, as well as a precursor to glucose. Okay. And I hope you found this helpful. Good luck studying for your test.